Okay, well, thanks everybody for sticking around to the end of the session. Today I'll be talking about evolutionary rates and disparity in the carnivoran cranium. This is work I've been doing with Jeroen Smaers, Christophe Soligo, and David Polly. And although I'm talking about rates and disparity, the framework that I'll be talking about these changes in is the framework of phenotypic integration. And we've heard a fair bit, actually, about integration and modularity already in this session, which is really nice, as I'll mainly skip this intro then. But the basic idea is just that evolutionary changes that occur in individual traits do not occur independently of changes in other traits. And this is something that we want to take into account when we're trying to reconstruct the evolution of traits or of organisms. And through quantitative analyses of morphometric data or indeed discrete data, we can identify highly correlated sets of traits. And in fact, these sets of traits can often form groups that have very strong interactions or correlations within them, but relatively weak interactions between them, and we call those modules. So this is just a little schematic. You can see that we can either treat morphologies as completely independent of each other. We can test to see if they are actually highly integrated, but we usually find something in between where we have these modules. And what I'm going to talk about today is actually not how we identify these or what they represent, but why we should even care about them. And the reason I think we should care is because trait covariances have been hypothesized very often to be a primary influence on morphological variation and individual character evolution. Okay, so I'll be showing a series of analyses that are based in large part on morphological data that I've collected over more than the last decade now. Couple thousand species that represent over 100, couple thousand specimens that represent over 100 species. These are 3D landmarks that are aligned using the standard methods. We generate trait correlation matrices for these and then subject them to various exploratory and confirmatory methods to identify patterns of integration and modularity. This is nice. We can do this with extant taxa, but we can also do this for well-sampled fossil taxa. And I talk about carnivorans a lot because they have a really nice fossil record. It means that we can actually apply these methods to things going into the Eocene, like Hesperocyon. So I've done this for a number of taxa across carnivorans as well as other mammal groups. And what we generally find, and again, this is long published, so I'm just going to fly through this just to set up the talk. But what we generally find is that within therian mammals, the marsupials and the placentals, we have actually relative conservation of cranial modularity. We tend to find there are 6 modules in the skull that have very strong correlations within them and relatively weak correlations across them. Of course, it's hierarchical. Monotremes, on the other hand, show quite a different pattern of modularity that's significantly different to what we see in therians. And ideally, what we would like to do is take these differences and say, okay, how does this translate into diversity? But unfortunately, the only clade that has a very different pattern has very few species in it, so we can't really set it up that way. So what I'm going to do today is show you ways I've tried to get around this problem. I'm not going to talk about limbs, but limbs are actually also very interesting in terms of their patterns of integration and show relative conservation, again, across large clades, but really nice differences across the mammal groups. And that's a promising avenue for future research as well. Okay, so the main question of this talk is whether or not modularity and phenotypic integration more broadly is evolutionarily significant. And like I said, we have these 2 basic divisions in mammals in terms of the cranial modules that we see. We have this monotreme pattern, where we have really strong correlations in the face and in the cranial base, but relatively weak structure across the rest of the skull. We don't really see clear modules within the rest of the skull, a relatively low level of integration. In contrast, across therian mammals, we see 6 modules, but there is actual variation in these modules in terms of the strength of the correlations within them. So we have 3 modules. This is the anterior face, the molar palatal region, and the basic cranium that have really quite strong correlations among them. And then these parts of the skull, the zygomatic, the orbit, and the vault actually have relatively weak interactions. That varies a bit across taxa, but that's a generalization. So using these differences, I want to try and understand how these patterns can affect morphological variation and long-term patterns of evolution, and really address the specific hypothesis of whether these patterns of trait integration constrain or facilitate morphological change. And those are kind of the end members of the possibilities for what the long-term effect of integration could be. Now, people have looked at this recently on what I would call a microevolutionary scale, looking at how trait integration responds to selection in various simulations, and also looking a little bit at data from Drosophila. And basically, what we do here is we take our covariance matrix that we get from some empirical data set, and we apply selection vectors to this covariance matrix and measure the response. And we measure a number of different attributes, but the ones I'll be talking about just very briefly right now are respondability, which is the total magnitude of the response to selection by this matrix that represents a population or a species. We look at evolvability, which is the magnitude of the response only in the direction of selection. And then we can look at a couple of angles. We look at constraint, which is the angle between the response vector and PC1 of your covariance matrix. So that, I would call that your path of least resistance. It's the vector that has the maximum variation in your real data set. And how closely the response vector fits to PC1 is a measure of constraint. On the other side, we have flexibility, which is how similar that response vector is to your selection vector. So what I've done here is take 97 of my covariance matrices and apply a series of 1,000 random skewers or selection vectors to these data sets, and measured these different attributes in comparison to what's right here, which is eigenvalue standard deviation. That's just a measure of degree of integration. So the higher the standard deviation of the eigenvalues, the more integrated your skull is. And what we see is that respondability, or the total magnitude of the response to selection, is highly correlated with how integrated your skull is. So the more integrated you are, the more you can respond to selection. But we find that it's very constrained in that that actual response will be aligned very closely to PC1 of your data set, the more integrated you are. In contrast, the amount of change in the direction of selection, only in the direction of selection, is actually not strongly correlated with integration. And what it really reflects is the angle between PC1 and the selection vector. And actually, when we look at flexibility, we find there's a strong negative correlation with integration. Here, I've put it against constraint. But basically, the more integrated your skull is, the less able it is to respond in the direction of selection. It'll channel the change along PC1 or along one of the major components of variation in the data set. So what this shows overall is that phenotypic integration does shape both the magnitude and the direction of response to selection by coordinating the response in the path of least resistance. Now, what I want to know is how does this scale up? This is essentially individual changes. But if we look at this on a large time scale, how can we scale up these results? And can we come up with hypotheses for what we would expect phenotypic integration to do to morphological disparity on large time scales? So first I'm going to show you some simulations on large time scales of the effects of integration and disparity. I'm using the carnivoran cranium as basically a sample data set. And then I'm also going to look at some of the empirical analyses that we've done, looking at whether modules of different magnitudes, so these very strong versus these very weak modules, show different disparities and different rates of evolution. Okay, so for that first part, we basically come up with a relatively simple simulation. It's a random walk model using a carnivoran tree with 36 tips in this example. It's 51 3D landmarks. And we've constrained the rates of evolution for each of the landmarks to the landmark variance from our actual data set. We rerun this 1,000 times, starting with the same shape. And we've run one with real trait covariances and one without any trait covariances at all. And I've done this for a range of 10 different covariance or correlation matrices. And these are just some of the ones that we've included. So something like a carnivoran matrix and something like the monetary matrix, as well as a range of them. And this, unsurprisingly to those of you who thought about this, is basically what you come up with. In red are the simulated correlated traits, and in black are the random traits that can do whatever they want. And this is for a sort of weakly integrated skull. This is for a highly integrated skull. And basically, the effect is that correlated traits, as you would expect, repartition the variance along preferred trajectories. So if you look at total morphospace, it looks like kind of uncorrelated traits can explore more of the possible morphospace. That's true. But actually, when we look at measures of variance-based measures of disparity, they're relatively similar between the two. Interestingly, though, when we look at range-based metrics of disparity, so in this case, maximum pairwise dissimilarity, we find that the integrated traits show much larger ranges than uncorrelated traits. And this is, so you can see in this right here, where we're looking at degree of integration versus the difference in the range of the correlated traits and the uncorrelated traits. The more integrated you are, the more extremal morphologies you'll basically be able to evolve. And so integration promotes the evolution of these extremal morphologies by partitioning the variance in preferred directions of shape change. And I think this will also have the likely effect, because it's forcing shape change along relatively narrow trajectories, it'll have the effect of increasing homoplasy and convergence among taxa with shared patterns of integration. Now we can think about what modularity actually does. I haven't demonstrated it here, but what modularity does is essentially break the variation into a few other axes of shape change. So probably it'll reduce the amount of extremal morphologies, but it'll allow the organism or the population to explore more of this kind of black morphospace. And I guess the way I think of it is like a big asterisk in the middle of a sphere, or sticking out from a sphere. And we can also plot this against time to try and better understand what the effect of trait covariances will be in the evolution of a clade. And in this case, the colors are swapped, but the uncorrelated traits are in red now, and the correlated traits are in black. And what we see is that each change for the correlated traits actually tends to have a larger range than what we see in the uncorrelated traits. So over time, while the mean morphology or the mean phenotypic distance from the original starting point is about the same, you have a much larger range in the correlated traits. And this can end up with things that look both far less disparate than your uncorrelated traits, but also much more disparate. So you have much larger range when you have trait integration. I think this also means that models of morphological evolution that ignore these attributes are going to have quite a bit of difficulty accurately reconstructing trait evolution. I'm actually going to just skip this because I'm running a bit over time, but I was trying to compare this to some of the empirical analyses where we show previously that looking at carnivoran data, there's relatively weak support for a strong influence of modularity on trait disparity, but the results that we do have tend to support a constraint model, although a little bit of that supports the facilitation model where trait integration increases disparity, and that actually kind of fits the expectations from the simulations. And then just briefly, I'm going to talk about how this affects, how integration might affect evolutionary rates. And what we've done here is taken a carnivoran tree and the carnivoran data set using an adaptive peak-based method of independent evolution, and there's the citations if you want to look at the specifics of the model. We've reconstructed rates of evolution. And this is just what we look at when we look at the entire data set, and you can look at clades, but I'm not interested in clades, I'm interested in landmarks, actually. And so what I've done is break this data set into the modules and looked at whether there are significant differences among the different modules in rates of evolution, and also how this relates to measures of disparity for those same modules. What we find, actually very surprisingly, is that there are some significant differences among the modules, but they don't correlate with either patterns of disparity in those modules, nor with module strength. So if we look at the oral nasal module and the basicranium are the most tightly integrated parts of the skull, and yet they have completely different, significantly different rates of evolution. So the basicranium actually has very high rates of evolution, whereas the facial region has relatively low rates of evolution. The vault is kind of a strange one because it has this one really big outlier that reflects the development of a sagittal crest. And my way of thinking about this, this is also just to show, this is the relationship between evolutionary rates and landmark variance. You can see there isn't a very strong correlation across the entire cranium, regardless of whether you exclude that one outlier or not. And so my way of thinking about this is that what modularity or trait integration does evolutionarily is it kind of creates a tube of preferred direction of shape change. But within that tube, things can evolve as quickly or slowly as they want to. A fly in a tube, I think, would be a fitting metaphor for what trait integration does to trait evolution. Okay, so just to conclude here then, Integration influences both the trajectory and magnitude of response to selection by directing evolution along these paths of least resistance. And we can estimate using things like principal component axes. And this can affect, on a large scale, can create both less diverse organisms, but also more diverse organisms than we would expect under kind of a random walk model. And I think this effect can be expected to produce homoplasia and convergent evolution more than we would expect randomly. In contrast, rates of evolution don't appear to be strongly influenced by morphological integration, and they actually show relatively little relationship to morphological disparity of individual modules, which I was quite surprised about. And again, this suggests that integration may shape the direction of evolutionary change, but not the speed of it. Nonetheless, though, I do think integration is problematic for morphological clocks and other sorts of evolutionary models, because the accuracy of those models depends not just on the variance in the rates of evolution, but also the number of independently evolving characters on which you're basing your estimate. So if we're building models where we're treating a lot of traits as independent and not taking into account the correlations among them, that's probably going to lead to inaccurate results. Okay, so thanks to many people who I've discussed various aspects of this research with, and various funding agencies, especially NERC and the NSF. And thank you.